This presentation is brought to you by Odo Achievement Center, a place for teens. Today we're going to discuss coronavirus. I'm your presenter, Dr. Banks. I'm a clinical laboratory scientist in New York City. I specialize in epidemiology and microbiology. Today, you're going to learn about understanding a brief history on coronavirus and how they are classified. We're going to distinguish between living and viral taxonomy. We're going to understand the mode of infection and transmission of coronavirus, which is called SARS-CoV-2. And lastly, we're going to identify effective ways to prevent the spread of COVID-19 in one's community using an online platform. So let's begin with the brief history of coronavirus. First and foremost, coronavirus was first characterized in 1965. It was discovered by two scientists named Dr. Tyrrell and Rino. Both of them discovered or noticed that some of the students at their college was getting respiratory infection, also known as a common cold. And upon closer look, they realized that this cold was attributed to similar viruses, not the same virus, but viruses that were very similar. And when we say very similar, we're looking at genetics, right? We're saying like two sisters are similar because they may share the same mother or the same father. But they're not the same. If they were the same, then they would be twins. So what they learned from their studies was that the viruses were similar, but they were not the same. They looked at the viruses under an electron microscope and determined that although they were similar and weren't the same, they all shared one common similarity. The surface of the virus had crown-like projectiles. And so the scientists decided to name the viruses that they saw corona, because in Latin, corona means crown, and they felt when they looked at the microorganisms or the viruses under the microscope that it had a crown-like appearance. So this term coronavirus was widely accepted in the late 1960s, and we now know that coronaviruses are common respiratory infections and have been seen all around the world since 1965. So how did they classify these viruses? How do we determine it was going to be coronavirus, and how do the scientists after 1965 associate other species of corona with coronavirus family? Well, to understand that, we first have to understand how organisms are classified. So we have living organisms, and then we have non-living entities. You and me, we're living organisms. Dogs and cats, they're living. Microorganisms like bacteria are also living. So we use something called a taxonomy. Taxonomy is how we classify and name organisms. And the naming of organisms is called using the nomenclature. So for taxonomy here, if you look at the left chart, under taxon, you have kingdom, phylum, class, order, family, genus, and species. And this is how organisms are classified based on these categories. The top category, the kingdom, is the most voluminous. It's the category with the most species. There are 5 types of kingdoms that we know of: plant, animal, protista, Monera, and fungi. 5. So the kingdom that I'm going to talk about today is the animal kingdom, which we call Animalia. Now I have 3 different species up here just so we can get an idea of how this taxonomy work. Well, as I said, the kingdoms are the most largest. And then from there, we have to go down a notch and further categorize these species. So phylum is the next one. Phylum says that all of these 3 species are chordata, meaning that they have a vertebrae. Class is the next one. And all of these species are mammalian. Mammalian means that they're able to conceive offspring internally in a womb, and the gestation process or the pregnancy is happening internally, and they all give birth. So species that have external gestational periods would not be classified as mammalian. The next one is order, and this is where you start to see the differences in the species and how we classify each species and name them and order them. Well, humans are considered primates, whereas dogs are considered carnivorous, and as well as cats. They're considered carnivorous because they have teeth that can bite into flesh. Primates don't have that. The next category is family, and now you see more differentiation. Family for humans is Hominidae. For dogs is Canidae, and for cats is Felidae. That's where we start to get the word feline. Genus is important because genus is basically going to tell us in what category these species are going to fall in. Humans is Homo, dogs is Canis, cats is Felis. Now, species is super important because now we're starting to speciate. We're separating them from their kingdoms, and they have now their own personal categorization. And each organism has this. So for humans, our species is sapien. For dogs, is familiaris. For cats, domesticus. So in the scientific world, when we're calling a human, we use the binomial name. The binomial name is the genus and species name. So in papers, we would write Homo sapien when we're referring to humans. For dogs, we'll write Canis familiaris. And for cats, Felis domesticus. So this is how we, in the scientific community, name organisms. Now, some of you might say, well, where did dog, cat, and human come from? Well, that's the common name. And that's going to be super important when we start to understand viruses. So the common name for Homo sapiens is human. Okay. So moving on to non-living entities. Viruses are categorized in microbiology because they're really, really small and they're really tiny to see. And in order to see them, you need to use an electron microscope. However, the word microbiology is confusing because micro means small or tiny, and biology means the study of living. But here's where we have a contradiction in the science. Viruses, all viruses, are non-living. They're non-living entities. They don't breathe, they don't eat, they don't sleep, they don't do anything that living organisms do. They can't even reproduce. So some of you might be saying, well, how do they infect someone? And we're going to talk about that a little later. But they undergo a process of multiplication, or what we say replication. And we'll talk about that a little later. But right now, I want to show you how we classify and name viruses. So we start off with order. They don't have a kingdom or a phylum or a class like the living organisms. We start off with order. So for coronavirus, the order is Nidovirales. The family is Coronaviridae. The genus is Coronavirus. So understand the genus is a large group of organisms or of a large group of viruses. And then from genus, now we have species. And this latest species is SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. And that's the species name. Now, the common name for SARS-CoV-2 is COVID-19. And COVID-19 basically means coronavirus 2019. And that's the common name. So now that you understand how we classify viruses, now we understand that coronaviruses is not something new in terms of 2019. It was first discovered in 1965. And based on the taxonomy, that group of viruses has grown exponentially because scientists were able to discover and test its genetic makeup using DNA sequencing to determine how similar they are, how different they are, etc. So now look where we are today with the Coronavirus family. As you can see, the chart on your right shows all of the coronavirus species that we know of today. That's a very extensive list that has been growing since 1965. Now, what we're going to add to this list, COVID-19. COVID-19 is very similar to all of these corona species. However, in the genetic coding, it has slight variations that make it different but similar to a previous outbreak that the world experienced, which is SARS-CoV-1. That was the last SARS epidemic that happened in 2003. In 2003, China came under a respiratory infection that infected a lot of their people and killed a lot of their people as well, similar to what's happening now. That was the SARS epidemic of 2002 to 2003. It did not reach the United States because we put in provisions to make sure that no one that was affected came into our shores. So we did a very good job of quarantining the United States and safeguarding the citizens of the United States from getting SARS. So now, with genetic testing, we know that SARS-CoV-2, aka coronavirus 19 or aka COVID-19, is very similar to the previous SARS-CoV-1. And if you look at the illustration on the left, you'll see how beautiful these non-living entities are. The red markers are the viral receptors, or we call them the viral spikes. They actually latch on to human cells or animal cells to invade and infect their host. Now let's talk a little bit about the mode of transmission. We have direct contact and indirect contact. Direct contact means person to person, means that you engaged in some way with the person that was infected. For example, coughing. Coughing goes 3 feet. So when someone coughs, the saliva and the droplets that come out of their oral cavity is spewing out 3 feet around them in a projectile-like manner as shown in the picture below. A sneeze will go 6 feet. So these are ways that the virus is transmitted, but the coughing or the sneezing person must have the disease in order to transmit it. If they don't have the disease, then they cannot transmit the coronavirus. So I want to make that abundantly clear. Another way scientists are just discovering is fecal oral contamination. They found that the coronavirus, COVID-19, is in feces. So when someone uses the bathroom, Number two, and they forget to wash their hands, now the virus particles are on their hands. And anything they touch can also become contaminated. If they're cooking food, it could go into the food. If they're making salad and they're tossing salad with their hands, now the virus is in the salad. And if someone eats that, now they're also contaminating themselves with COVID-19. Indirect contact is basically touching, or touching something and then touching your eyes and your mouth. So here's a chart from the New England Journal of Medicine that was put together by Business Insider. And they basically truncated the information to a nice picture. So in air, when someone sneezes or coughs and they are infected with coronavirus, it can last, those aerosols can last in the air from anywhere between 30 minutes to 3 hours. Change, money, coins, it can last on copper for 4 hours. So if someone coughed in their hands and then dug in their pocket and picked out some coins, and let's say they bought something at the store using those coins. Well, now the person that took the coins is now exposed to COVID-19. And if they don't wash their hands and they eat food or they rub their eyes, now they just expose themselves to the virus and allow the virus to now get into their body. Cardboard is 24 hours, stainless steel is 2 to 3 days, and plastics, like your plastic containers that you put food in, can hold the virus up to 3 days. So let's talk about mode of infection. So I like to coin this term APUSAR. APUSAR stands for A, absorption. And if you look at the top right-hand picture on the screen, you'll see that the spikes on the virus is connecting with the receptors of the cell. And that's called adsorption. And that's when the virus attaches to the cell, the receptors on the cell surface. Then from adsorption, you have penetration. And this is when the cell is going to receive the virus and allow it to penetrate inside of it. Uncoating is when the virus will release its enzymes, viral enzymes, to break down the coating that was created when it came into the cell. When it uncoats, it releases the nucleic acid. Now, coronavirus is an RNA virus, so it only has one strand, ribonucleic acid. So that one strand, that RNA, will then be released into the cytoplasm of the cell. And when it's in the cytoplasm of the cell, it's going to make its way over to the nucleus. The nucleus is the brain of the cell. It basically tells the cell exactly what it's going to do. It coordinates everything in regards to the cell. So the RNA from the COVID virus is now going to embed itself into the nucleus of the cell. And when it embeds itself into the nucleus of the cell, it basically hijacks the cell and say, hey, stop doing what you're doing and only transcribe me. Transcribe means to make a copy. So now the nucleus under hijack is now going to synthesize, or what that means to make or create, the viral proteins that is genetically coded in that RNA sequence. So this can be spikes, capsomeres, new RNA. So it's going to synthesize all of the viral proteins in its parts, not as one whole viron. The next thing after making these viral proteins, it needs to assemble. and it's going to assemble inside of the cell. And that's pictured number 5. So now, once it assembles, it's ready to be released from the cell. And coronavirus is an envelope virus. So when it releases itself, it's stealing part of the cell's cytoplasm and cell envelope. So this makes it really difficult because the immune system will believe that this newly released virus is now part of the body as opposed to being identified as a foreign entity. So what does this mean? Well, let's think of it as a police. Okay? The police is going around and they surveillance the neighborhood to make sure that there's no crime. And if there is crime, they stop it and they arrest the person and bring the person back to the police station to get them off the streets. Well, that's the same thing with what our immune system is doing. Our white blood cells is going around our bloodstream and our body to survey to make sure that there's no foreign entities that could potentially cause harm to us. If there is a potential entity that can cause harm, the white blood cells would arrest it and then take it back to the lymph nodes to destroy it. Well, what this virus is doing is pretending to be a cop. It's pretending to be a normal person. But really, it stole the identity of the cell. So the white blood cell will go up to it and say, okay, you're one of us. You're free to go. And then let that virus go to infect yet another cell. So the immune system has to be smart and wise up and be able to identify when the cell was hijacked and when the virus is pretending to be part of the body. So now that you understand how the infection works, let's talk a little bit about the chain of infection. So let's go over here to the right-hand side of the screen where it says germs. So anything can cause an infection, a bacteria, a parasite, but in this case, we're talking about a virus as a non-living entity, specifically COVID-19. What happens is the virus is then introduced to people, animals, food, soil, and water, and it makes the community of people are now susceptible or potentially exposed to the virus. So if someone actually gets sick, what happens is they start to emit the virus from a portal of exit. A portal of exit is like the mouth or a cut in the skin. In this case, for COVID-19, the portal of exit is feces and coughing or sneezing. So when the person coughs or sneezes, they're releasing the droplets, salivary droplets, and on the droplets, you'll have the coronavirus. This can be done, as we stated before, through indirect or direct contact. So that means if someone coughs in their hand and then touch the doorknob, and the doorknob is made of stainless steel, and someone else comes who doesn't have the disease, will touch the doorknob, and then rub their eye or eat food before washing their hands, now they've been exposed to it. That's a mode of transmission. And then from there, the person would have to introduce the virus into their portal of entry. A portal of entry is how the virus gets into the body. So portal of entry for coronavirus is the mouth or the eyes. So if somebody sneezed as a mode of transmission, and you're standing within 6 feet of that sneeze, and that's the droplets fly over to you and get into your eyes or your mouth, or you breathe it in through your nose, well, that's all portals of entry. However, if they sneezed and you turned away immediately and they sneezed on your back, and it did not get into your face, well, then it did not get into your port of entry. But let's be mindful. If you rubbed your back and then ate food before washing your hands, then you potentially expose yourself. So we have to think about these things, and we have to think critically about ways we can prevent the disease from further infecting others. So now, let's say the person was susceptible and they got it through their mouth. Now what happens is they're now contagious and could potentially infect other people. Now, when they infect other people, there are some people that are already sick. And if they're already sick and then they get introduced or exposed to the coronavirus, it makes their immune system extremely weak. So these are babies, small children, the elderly, people with weakened immune systems, or anyone can be susceptible to the disease. But those who are dying are usually those who have some type of pre-existing condition or they're elderly. And that's because their immune system is already fighting a war with their pre-existing condition, or they're already fighting a war because they're older. And so their immune system is not as strong as it used to be when they were younger. So it's so important that we safeguard everyone from getting this disease so that no one can die. So now I'm asking that you all meet me on Instagram, and we're going to have a live session on Instagram where we're going to talk about answering the question, how to prevent the spread of coronavirus in our community. What can you do in your home, with your family, and in your community to prevent the spread of coronavirus? So log on to our Instagram, or you can go to our Instagram and go right on to Zoom. Zoom, you'll be able to connect with me one-on-one. And we're going to have this open live session on Friday, March 27th, 2020, which is tomorrow, at 5:30 p.m. So be ready, be there, and I look forward to seeing you and answering your questions and just having a conversation about coronavirus.